Hello everyone, I am Ana Paula Stein Santos. It's a pleasure to be here to present you a part of my doctoral research titled The Role of the Mushroom Culture in CO2 and CH4 Balance. This work was conducted in the Federal University of Santa Catarina and University of Itajaí Valley in partnership with Embrapa. There is a great concern about the concentration increase of the GHG in the atmosphere, especially carbon dioxide and methane. However, the marine emissions have been poorly studied. The ocean waters, especially coast waters, are naturally rich in organic matter, becoming large accumulators and large carbon accumulators and potential GHG emitters. Intensive mariculture could be an increasing contributor to the organic matter inputs, and it is an important approach in the issue of global climate changes. Estuarine and resurgence areas have greater inputs of organic matter, which may lead the emissions of GHG by the mineralization of the organic matter. So, how it happens? Organic matter and nutrients enter in the water from the river inputs and provide foods from the phytoplankton. And then are sources of food from the filter mollusks. Mollusks returns this organic matter in the form of pseudofeces. These pseudofeces can be recycled in the water column or deposited in the sediment floor. In the upper sediment on oxygenated layers, the more labile fractions of the organic matter were degraded, using CO2 as electron acceptor. The most refractory organic matter percolates in the sediment, suffering the degradation by another electron acceptor, such as nitrate, manganese, iron, and sulfide. When this acceptor becomes exhausted and the sediment oxide, the methanogenesis starts, producing methane. In marine sediments, due to the high availability of sulfide, this sulfate reduction layer is very extensive, requiring a high concentration of organic matter to percolate and produce methane. The crops also are great importance in CO2 balance, since sometimes they act as the emitter by the respiration and the pseudophases degradation, and sometimes as a receptor by the absorption of CO2 to the formation of carapaces, or by ingested the organic matter by the phytoplankton. Food industry is the major contributor to the GHG increasing, especially cattle, as we can see. Other crops, although in a lesser extent, have higher CO2 emissions also. So in this point of view, mariculture can be a sustainable animal source of protein. In addition, mussel and oyster can act in the CO2 sequestration by the biological balance and the shell formation, as we see in the figure. And it is important because the aquaculture system has grown faster with the overexploitation of marine fishing. Since 1999, we have a stability of fishing resource. Even with fishing technology increase, there's no more biomass to supply the demand. Until now, the most crops of aquaculture are land-based, but the marine crops have been growing in the recent decades. And the aim of our work is evaluate the influence of mussel production on CO2 and CH4 emission in the coastal region of southern Brazil. The study area is located in Armação de Itapuporó Inlet, municipality of Penha, state of Santa Catarina, Brazil. The area was divided in Beach Point, here near shore, mariculture site inside the mussel perna crops, and control site outside, outer, and without mariculture. The study was conducted for 3 days for the greenhouses collection. Carbon dioxide and methane fluxes were determined by statistic chamber techniques using 1 liter cylindrical chambers, like this in the picture. The fluxes consist in the periodic samples from 1 chamber every 7 minutes, one collected. The air collected was analyzed by gas chromatography, and water dissolved and air ambient concentration was also determined to do corrections. In situ parameters was carried by Oriba multiparameter probe, and water sample from the water column in subsurface were collected by Niske bottle, like this, and storage refrigerated in plastic bottles until processing. Water quality parameters were determined from standard method of examination of water and wastewater, as we see in the shown table. So as a result, the study area has an average salinity of 28.9. The oxygen concentration vary between 7 and 8. What is expected for the area? With a slightly lower concentration in mariculture site, possibly due to the breathing process by the mussels. Suspended particulate matter has shown the same behavior, with a subtly lower concentration inside the mariculture crops, which may be related to the reduction of hydrodynamics by the presence of mussel long lines. Has the same turbidity, showing highest concentration in the shoreline sites near beach, what is expected due to the resuspension of sediment by the waves. pH has a small variation inside the points, and present value ideal for the mariculture activity. The average concentration of nitrate was 1 microliter, and represents a little part of the ammonium concentration. This pattern is common in environments with recent input of nitrogen compounds, such as input of effluents, as an example. We can see a slightly increase of ammonia inside the mariculture areas that can indicate a source of this nutrient by extraction by the bivalves. Chlorophyll showed low concentration in mariculture sites, which may indicate the consumption of phytoplankton by the mollusks. Total organic phosphorus and phosphate has no difference between sites, but phosphate showed the highest concentration in control points, which can be related to are quickly consumed by this element for the organism, due to this chemical reactivity. This study showed the low methane emission when compared to other type of carbs, as was expected. The methane fluxes was higher in control sites, suggesting that the mushroom culture did not influence on methane emissions. These highest emissions in the control sites could be related with the hydrodynamics and another environmental factors that should be further researched. Carbon dioxide show also negative and positive values of flux, and carbon sequestration also occurred in control and mushroom farm sites, and it didn't show significant difference between the areas, suggesting that the mushroom culture did not influence on CO2 emissions. Any parameters showed high correlation with the emission of the GAG. However, especially for CH4, dissolved oxygen in nitrate showed a significantly low positive correlation. So our preliminary results revealed that the mushroom farming in the sampling site did not influence significantly the CO2 with BO4 emissions. So results showed that the mushroom farming in Tapukura Inlet did not have an environmental impact in GAG emissions. It means that the flux were similar in all points, what indicates that the production of mushrooms did not affect the natural GAG emission in the study area. A few monitored parameters have no difference between control areas and mushroom areas, showing no impact in results from the mariculture activity. And all parameters show the environmental health levels, indicate no impact on the water quality and greenhouse emissions. As a recommendation, further research in marine aquaculture parks, improve analytical tools, and continuous monitoring. I like to thank you for the attention, and thank you all the institutions involved in this project and the SEMAS conference by the opportunity. Thank you.